Hi, my name is Nachiket.
My name's Gino.
My name's Howie.
I'm Shalky.
And my name is Tashi.
And welcome to our presentation on the digestion of Ursus maritimus. Our group was tasked with investigating the digestion of thermoregulators, and in this presentation, we will go over the adaptations of our animal, thermoregulation as a whole, the digestive system, details into our two experiments, and finally our discussion and conclusion.
Ursus maritimus, or commonly known as the polar bear, is perhaps one of the most recognized animals of the Arctic, if not the world. A member of the class Mammalia, polar bears share many characteristics with the other mammalian species. These characteristics include fur, mammary glands, and the ability to regulate their internal body temperature. More specifically, as a member of the family Ursidae, which dates back millions of years, polar bears share their evolutionary history with the likes of 7 more extant species. These species are polar bears, black bears, brown bears, the giant panda, sun bears, sloth bears, and spectacled bears. Going further down the taxonomic classification under the genus Ursus, the black, brown, and polar bears make a monophyletic clade, with the polar bears and brown bears having only speciated within the past 6 million years. However, even though they only recently diverged, the frigid cold of the Arctic provided sufficient selective pressures that divergence was rapid.
So as polar bears diverge into their own species, their internal systems, adaptations, and physical attributes start to differ drastically to maintain fitness in their new environment. They have small ears and a short tail to help reduce heat loss. They have 2 layers of thick fur and black skin to help absorb heat. They have adipose deposits under the skin, which helps with insulation and serves as a store of fat reserve. They have specialized front paws for swimming, which can be utilized when hunting for their prey. Lack of fur pigmentation helps in camouflaging in their icy habitat. And polar bears have shorter, sharper, and more curved claws, which helps with easier movements on icy surfaces and having a better grip on their prey. As polar bears are mainly carnivorous because of their habitat, this resulted in cranio-dental adaptations like sharpening of the molars and a gap between the canines and molars, which allowed deeper canine penetration in their prey. So for the behavioral adaptations, we can look at their diet and gradual processes like hibernation. They are the most carnivorous of the extant species of bears, but they do feed on vegetation during the ice-free season. They have a lipid-rich diet that consists exclusively of marine mammals such as ringed seals. They have a lipid-rich diet that consists exclusively of marine mammals such as ringed seals. They need the blubber from the ringed seals as it provides with the nutrients they need to stay warm in their extremely cold habitat. And the polar bears' hunting and eating patterns depend completely on sea ice. When it comes to hibernation, polar bears have specialized winter dormancy where only pregnant polar bears hibernate.
Polar bears are thermoregulators. Specifically, they're endotherms, which means that they have the ability to maintain a steady internal body temperature, irrespective of their external temperature. There are several physiological mechanisms which allow endotherms to generate a steady body temperature. One such way includes shivering, where the organism can quickly contract the muscles to generate friction and ultimately heat. Another mechanism is through increased metabolism, where a greater number of mitochondria present in the cells of the organism allows for more ATP production, which results in more metabolic heat production. There are also several mechanisms whereby endotherms can conserve this generated heat, such as having a larger body size, which is exploiting the square cube law by having a larger body volume inside a smaller body surface area. Furthermore, they have evolved fur or hair, vasoconstriction, and insulation through a fat or blubber layer to help conserve more heat.
Digestion is the main process through which living things gain nutrition and maintain their health and well-being. Food availability has recently gained an increasing amount of attention as populations and economies continue to grow globally. However, it's important to dive deeper and understand digestion on a physiological level, as it may bring rise to important aspects of consumption that are not evident at first glance. In humans, the digestive tract can be split up into 4 components that interact in a series formation. The first is the mouth, where oral processing occurs, followed by the stomach, where gastric processing occurs. Next is the small intestine, which is the main site of intestinal processing, before arriving at the final destination of the large intestine, where fermentation is carried out. In contrast to humans, polar bears are massive in size. Therefore, it's safe to assume that they require massive amounts of energy. Interestingly enough, there's limited information in regard to how polar bear digestive systems work. In contrast, black bears have been mentioned in scientific literature for decades on topics ranging from the digestive system to their reproduction rates. As mentioned earlier, black bears and polar bears experienced a recent diverging event. In regard to cladistics, this serves as valid grounds for sound comparisons to be made across the 2 groups. Black bears have digestive tracts that are short, forcing them to rely on the muscular lining of their stomach for a majority of food breakdown. Once food is consumed, it follows a path similar to the one found in humans, traveling down the esophagus and into the stomach where chemical digestion occurs. The mixture then moves into the small intestine where additional disintegration occurs before the components are absorbed into the bloodstream. The lack of scientific data, combined with the recent divergence, allows us to assume that polar bears exhibit digestive functions that are similar to that of black bears. However, one thing to keep in mind is the difference in diets that exist between the 2. While black bears concentrate more on consuming plants, polar bears feast on marine mammals. Polar bears can digest protein and fat at efficiencies as high as 84% and 7% respectively. This serves as a reminder to consider the varying constraints placed on the digestive systems of different bears when using one as a reference point for the other.
So this slide shows the seasonal timeline of polar bears. So during the wintertime, polar bears, they do not hibernate because it's the most important time of the year for them, as they do majority of the eating during winter. And only pregnant female polar bears den up during the winter, but the others do not hibernate like brown bears and black bears. During the winter months, when the sea ice surface is present, it is prime seal pupping season, which means it is prime hunting season for polar bears. This season is very important for them to hunt and store as much fat as possible as they need it for the summer. So, and then for the springtime, climate change is causing ice to melt earlier in the spring, and this increasingly limits the time polar bears have to hunt for seal pups, from which they historically have gained the majority of their annual fat reserves. As sea ice begins to crack and melt, most bears begin to return closer to shorelines. And for mother polar bears and cubs, this is a beautiful time of the year where they will emerge from their dens around March and early April and are introduced to the beautiful world outside. While mothers are occupied with the cubs, early spring is also a prime time for the mating season. And for the summertime, they mainly live off their stored adipose tissue, which they obtain from hunting during the winter season, and opportunistically feed on various marine or terrestrial based diets. And for the fall, it's a very exciting time for the polar bears as they wait for the water to freeze so they can start hunting.
So what will we be investigating? We will look at 2 hypothetical studies exploring how temperature ultimately influences digestion of polar bears. So much research has been conducted on how polar bears have adapted to live in the harsh Arctic ecosystem, but not enough research was found which attempted to test how differences in external temperatures can influence the physiology of the polar bear. Thus, in this study, my team and I attempted to test how changes in external temperatures can influence the digestive physiology of the polar bear. Specifically, 2 experiments were conducted as mentioned previously. The first experiment, whose purpose is on this slide, ultimately attempted to test how changes in the external temperature can potentially influence the digestive efficiency of the polar bear in regards to certain macromolecules, namely lipids and proteins. As such, the null hypothesis for this experiment was that the digestive system of the polar bear did not show a significant difference in digestive efficiency between consumption of different macromolecules in the standardized diet that will be provided to the bears. On the other hand, the alternative hypothesis was that the digestive system of the polar bears did show a significant difference in digestive efficiency between consumption of different macromolecules when provided in their standardized diets.
The purpose of experiment 2 was to design an experiment which tests for microbe diversity that was present in the polar bear fecal matter, and that would provide us information on their digestional changes, which are affected by changes in their diet that would occur with seasonal temperature variations.
So the null hypothesis for experiment 2 states that no changes in gut biodiversity are expected based on temperature changes throughout the year. On the other hand, the alternate hypothesis states that as the environment warms over time, the digestion rates of polar bears will decrease.
Experiment 1 explores digestive efficiency of lipid, protein, and berries using indigestible markers. This study acquires 3 female polar bears from the Hudson to perform digestive analysis. The polar bears are 2.4 to 3 meters in length and weigh approximately 450 pounds, found in the same geographical area. To maintain an ideal experimental sample on temperature and digestion, the polar bears need to be acclimatized to the same climate and diet before the start of the experimental process. The sample group is standardized in separate rooms with the ambient warm temperature of negative 15 degrees Celsius, a standardized diet with exactly 150 pounds of food, which contains 90% seal and 10% berries, is provided throughout a series of 43 days, with 1 eating day, which is followed by 5 rest days, for a total of 8 eating days. The food is grounded to a homogeneous state with a commercial grinder to minimize selectivity of the food. They should be provided adequate space and habitat features to maintain a close relativity to the everyday environment of a polar bear. When conducting the actual experiment, the polar bears are stationed into 3 different temperatures, as previously mentioned: cold, which is negative 35 degrees Celsius, hot, which is negative 6 degrees Celsius, and the control, which is negative 15 degrees Celsius. 2 days after the last diet, during standardization, each polar bear will be provided a control diet of berries, which contains 50% of the berries that are marked with chromic quoxide, and the other 50% will be marked with ytterbium chloride. These are indigestible markers. The fecal matter after the berry consumption will be collected and observed. 2 days after that, they will be provided with the experimental diet. The diet provided consists of the standardized quantity of 150 pounds of food, but is altered in terms of composition. So 45% of it will be protein, which is muscle, 45% of it will be blubber, which is fat, and 10% will be berries. An indigestible marker, ytterbium chloride, is added to the protein component of the diet and thoroughly centrifuged to assure marker protein binding. Moreover, the lipid or the blubber content is centrifuged with another indigestible marker called chromic quoxide. The diet will be provided with 5 days of rest for a total of 12 feeding days. After each feeding day, the fecal matter is collected, freeze-dried, and circulated in a Willy mill with a 667 RPM. This fecal matter is then analyzed for the number of ytterbium chloride and chromic quoxide markers present in the fecal matter using an atomic absorption spectrometer.
So this slide shows us the expected results for the first experiment. And as you can see, each macromolecule and its associated marker is shown as the column headers, whereas the percentage of the markers that are retained in the fecal matter are shown on the row headers for the treatment and control groups. Starting off with measuring lipid digestibility, the treatment bears show an arbitrary retention percentage in relation to the control bear result. The colder treatment bear sees a decrease in percent retention, whereas the warmer treatment bear sees an increase in retention. When looking at protein digestibility, there isn't a significant change in percent retention of the ytterbium markers. And finally, sorry. So you don't see much any percent change in the ytterbium markers between the control and the treatment pairs, which is different than what was seen for the lipid digestion markers. And finally, the last two columns that are seen in this table are for the berries for both the markers. The expected result for the berries for both markers are 100% retention. And this slide just shows us the graphical representation of the table that was seen on the previous slide, where the x-axis highlights the treatment and control groups, and the y-axis shows us the percent change in digestive efficiency for lipids, proteins, and the berries. So once again, the colder treatment bear should show a decrease in percent retention of the lipid chromic markers, and the warmer treatment bear should show an increase in percent retention of the markers when compared to the control treatment result. There should be no significant change in the percent retention of the protein markers between the treatment and the control bears. And finally, 100% of the markers are expected to be retained for the berries for both markers across all three bears in each treatment and control group. So this slide just shows us as an overall statement what is expected to see for the lipid result. And this slide is showing us the expected result for the protein marker digestibility.
Berries are added in this experiment as the positive control. Berries contain cellulose, a macromolecule that is indigestible by all mammals. The reason why mammals cannot digest cellulose directly is because the genes for the particular enzyme needed for the breakdown of the beta-acetyl linkages are not present in mammalian genomes. Herbivores can break down plant products, and thus cellulose, due to the symbiotic bacteria present in their gut, but carnivores do not have these bacteria present either. Thus, when the polar bears are provided berries, the cellulose present should not be digested, and 100% of the markers on the berries should be retained in the fecal matter. This should highlight that the markers are indeed present on the macromolecules and that they are functioning. The reason why the colder treatment bear is expected to see a decrease in percent of lipid markers in the fecal matter is because of digestive efficiency is expected to increase when the bear is placed in colder external temperatures. As mentioned previously, one of the ways that endoderms retain body heat is through a thick fat or blubber layer. These layers conserve a thermal insulative function through two mechanisms. The first is through a physics concept. So heat, as all forms of energy, tends to dissipate and expand through the second law of thermodynamics. Thus, having a thicker fatty layer would increase the distance the heat would need to travel, and as the traveling distance increases, the less heat is ultimately lost. In addition, fatty layers also contain a lot more blood vessels than normal tissue, so the effects of vasoconstriction on thermal insulation are more prevalent. Both of these effects are increased with a thicker fatty layer, and as such, it is expected that in the colder temperatures, polar bears will gain this thicker layer. In order to do this, the lipids that the bears eat need to be more efficiently digested, and hence, the expected results of a lower percent retention of the indigestible markers should be observed.
So proteins, on the other hand, are not expected to show much of a change in digestive efficiency in regard to external temperatures, as was seen in the table and the figure. So proteins definitely serve several functions for a mammal, ranging from biochemical messengers in the form of hormones to cell growth and repair, where all sorts of transcription factors and polymerization molecules are present, and even structural integrity of cells and tissues, for example, the protein collagen. However, proteins have not been shown to directly influence heat generation or retention as lipids do. As such, we do not expect the digestive efficiency of proteins to change too much when the bears are exposed to extreme external temperatures on both ends, either the cold or the warmer treatments. However, the indirect effects of temperature changes on protein functionality and digestibility have not been tested. For clarification, though the digestibility of proteins may not be directly affected based on an adaptive reason. For example, there's no need to better your digestibility of proteins as compared to lipids for thermoregulations, because lipids directly provide you a survival adaptation in the form of your adipose layer, whereas proteins do not have such an adaptive reason for it to be for its digestibility to be increased. So even though there may not be a direct reason for this, there may be an indirect influence due to other factors. So for instance, the breakdown of lipids and proteins both alike are conducted through enzymes such as lipases and proteases. And these enzymes are susceptible to temperature changes, and they can denature if the internal body temperature of the polar bear is outside of the enzyme's comfort zone, for a lack of better words. So essentially, if enough enzymes denature in the polar bear, this can result in changes in digestibility because there's simply not enough enzymes to saturate and break down these macromolecules. And this is definitely something that should be considered for future tests. In addition, the composition of the fat layer and the changes that are made to the fat layer can also be studied. A paper had recently demonstrated that based on what polar bears are fed, the fatty layer of the bears differed between each other. In addition, the adipose layer, which contains a total of 3 layers, the inner, the middle, and the outer layer, also differed from each other respectively and with each other in respect to other bears. So the outer layer of one bear would differ from the outer layer of another bear based on what the bears were fed. And in this study, the bears were strictly fed ringed seals. So for future studies, it is highly encouraged that different kinds of seals and even different kinds of blubber from other animals, such as walruses and beluga whales, should be fed to the bears, and changes in external temperatures can then be tested to see if lipid digestibility and storage are influenced.
So the second experiment will use fecal bacteria content to investigate the digestion efficiencies of polar bears. A group of polar bears were observed in their natural state from the beginning of one summer to the end of the winter over 3 consecutive years. To ensure the same population was being studied without disruption, a broad range of their habitat was marked off as valid study grounds, and observations were only made within these parameters. At the end of each day, fecal samples were collected for experimental observation of its bacterial composition. The feces samples were extracted using DNA mini kits. Amplification was then carried out through PCR, and the products were visualized on a 1.5% agarose gel. Following this, the reads were processed, and specific microbial levels in each sample were statistically analyzed through processing through the use of an R package. So the table on the right shows the expected level of bacterial operational taxonomic units, or OTUs, over the 3-year observation period, while the graph on the right is a visual representation of this. And as you can see, there's an obvious trend. The paper used in predicting these results was done by Watson and his colleagues, who observed a decrease in bacterial levels in the polar bear feces as temperatures decreased. In their study, they investigated the different content of bacterial OTUs in polar bears that lived onshore versus those that lived offshore. They found a higher amount of bacterial content in the feces of onshore bears who were subjected to altered habitat conditions caused by warming temperatures. In the offshore bears, they did find bacterial OTUs, but at much lower levels because they maintained a regular diet. So here's a bit more detail on the expected results. In year 1 of the proposed experiment, a gradual increase in bacterial content of the feces was observed from February to July. As seen in the table and figure on the previous slide, the OTU levels start off at a minimum of 62 and peak at 242 OTU. Following this, a decrease of levels began in August and continued up until the month of January, dropping from 213 OTUs to 70 OTUs until finally reaching the second starting point of observation. Year 2 OTU levels started at a minimum of 200 in February, gradually increasing to a high of 551 in July. In August, a decrease began to be observed at 485 OTUs that gradually declined to a value of 220 OTUs in January. In the third and final year of observations, the highest values of OTUs were observed. Levels grew from 343 in February to 860 in July. Similar to previous observations, in the month of August, a gradual decrease began, starting at 757 OTUs, and finally ending with 370 in January. These results show a gradual increase of bacterial OTUs when temperature begins to increase, but also as observational time goes by. The average low of OTUs goes from 62 in year 1 to 343 in year 3, while the average high grew from 242 OTUs to 860 OTUs.
The results shown here display the dietary changes only for the winter month. The figure shows the graph for the winter season where the 3 different food variables were tested. The 3 food variables being the blubber, muscle and viscera, and berries and plants, which were the limited food sources that are available to polar bears during the winter months. The trend which was seen for the winter season was the polar bears preferred to eat blubber the most, which is the fatty layer of the seal. Next, the second preferred option for the bears was the muscle and viscera, which is a protein component that the seals are composed of. Lastly, the food variable which the polar bears preferred the least among the 3 was the vegetation option that consisted of berries and plants that can be found in the tundra. To analyze the relationship seen in the previous figure, the comparison can be seen between blubber being eaten the most from the entire 3 variables presented to the polar bears, which can be explained through reasoning that the blubber is made up entirely of fat, which is what polar bears are able to store during the winter months for a chance at a longer survival, meaning that they have fat stores which they can use for longer periods of time before they have to go out and hunt again. Thus, the polar bear preference for blubber correlated along with its fecal component that was measured using the lipid marker, where it was seen that lipids which make up the blubber were tested for the highest content in the polar bear gut digestion. This may also correlate to why polar bears are seen to tear away seal skin in order to reach the blubber fat layer during the winter months, when they are not at risk for starvation because of lower temperatures, allowing for ice to remain as hunting ground. In addition, this also explains why the polar bears on a behavioral level would not opt for muscle and viscera, as the protein would be much more time-consuming to digest than fat content in the blubber. It would not benefit them as much as the fat consumption would in terms of energy obtained. As for vegetation, berries and plants had the least rate of consumption as they offered no fat or protein for the bear. One can also correlate that the winter months makes it difficult for the bears to scavenge for berries and plants on and offshore. Thus, seal hunting would provide a larger advantage and would be a more widely available food source during the winter months. Now, for the summer season results, the experimental results displayed that the most abundant part of the polar bear diet was the muscle and viscera. Closely following muscle and viscera was seen to be the consumption of blubber. And lastly, the least abundant or the least preferred food source for the polar bears was seen to be vegetation, once again being the berries and plants. To discuss now the summer months trends, the previous figure showed that there was a significant increase seen in the consumption rate for berries and plants, and a decrease was seen for blubber consumption, while muscle and viscera was at an all-time high. This may not mean that the polar bears prefer muscle and viscera over blubber during the summer months, but it more so refers to them not wasting any part of the seal because of their availability to capture them during the ice melting months have now decreased, making each part of the seal now a valuable source of food. Thus, because muscle and viscera make up more of the seal than the blubber does, it would correlate with why the results for muscle and viscera have increased all of a sudden during the summer season. Therefore, the experimental observation concludes that polar bears have a preferred affinity for fat content over protein, as all the figures display the trends for fat consumption being relatively higher overall for both summer and winter months, whereas protein would be their next best option at a food source. The results here display the OTUs corresponding to specific temperature changes. The table seen here shows June to be the month where the most bacterial OTUs were identified, which was 802 OTUs. The month ranging slightly before and after June display a decrease in the gut microbe diversity, for example, May showing 602 OTUs, July and August being in the range of 700 OTUs. However, for the spring months such as March, April, and May, a range of 400 to 600 OTUs were seen. Similarly, for fall months such as September, October, and November, a range between 700 to 500 was seen. However, when noticing the winter months for December, January, February, and the beginning of March, a steep decrease in the OTUs can be seen in comparison to the summer and fall months. Thus, the trend of lower OTUs correlates with months with the lowest measured temperatures, which were December, January, and February, all ranging with temperatures between -20 to -25 degrees Celsius. Whereas the months with the higher environmental temperatures measured were June, July, and August, all ranging for temperatures within 3 to 7 degrees Celsius. Furthermore, the OTU's abundance in the summer months also displayed that the phyla of the bacteria which were tested for, Firmicutes, Bacteroidetes, and Proteobacteria, were all present in the higher abundance in the polar bear gut during the summer months. In comparison to the winter months, the same bacteria were seen to be present at a much lower abundance. Thus, a pattern could be seen among warmer months, allowing for a diverse microbial gut in polar bears. When integrating this data with the first experiment, it can be observed that this may be because of summer months causing polar bears to have an increase in their diet range, which would mean a wide variety of food consumption, leading to a wider variety of microbes in their gut that would also allow for a much more efficient digestion rate. In contrast, the winter months can also be concluded to have the opposite effect on digestion rate efficiency, since a decrease was seen for the gut microbial rate in the feces for the months ranging from September to December, and then slowly increasing from January to May. The temperature can once again be compared to know whether temperature change had an effect on the microbial diversity or not. The lowest temperatures were observed during the winter months of December, January, and February, all of which experienced the lowest microbial diversity of 400 OTUs, 352 OTUs, and 382 OTUs, respectively. Furthermore, when examining what it means to have a higher diversity of these microbes from the phyla Firmicutes, Bacteroidetes, and Proteobacteria in the polar bear gut, they can be observed to have a function of allowing for the metabolism of undigested or harder to digest food, such as vegetation in this case, where the polar bears have no choice but to resort to in times of starvation during the summer months, when seals are no longer a fast hunt due to the melted ice grounds. Specifically, the presence of Bacteroidetes and Firmicutes allow for a higher metabolic efficiency in the gut, as their function is to digest fiber and polyphenols, which can be found in food remnants such as berries and Arctic vegetation, which we tested for in Experiment 1. If integrating the use of these gut bacteria on a molecular and species level, these gut bacteria can also be found in humans, playing the same function, which allow for an overall healthy gut environment by contributing to helping with defense and the immune system and maintaining overall normal function. Thus, the microbes allow for this efficiency through their energy harvesting mechanism, which allows for nutrients to be extracted through co-metabolism. In addition to gut bacteria having the function of allowing for a healthy digestion to occur, they also play a role in determining obesity. If observing a high-fat diet, which was seen in the winter months for polar bears, it would be assumed that a high-fat diet would allow for an increase in weight gain, which would be an evolutionary benefit for female polar bears who would hibernate during the winter months, because they would not have to worry about hunting for food, as they would be able to rely on their fat stores. So in terms of thermoregulation, they would be able to reserve heat through these fat stores in the cold Arctic environment. Thus, changes in gut bacteria occurring, such as a decrease during the winter months, can lead to a higher obesity rate in the polar bears, as they no longer have an efficient mechanism for digestion, thus decreasing their metabolism and allowing for most of their food to turn into fat stores, which are useful as a form of energy uptake and heat conservation during the cold winter months.
When discussing the role of microbes in digestion and gut health, we can conclude that they play an important part in providing essential metabolic functions, such as assisting with the immune system of the polar bears. The same can also be applied to human health if we compare species. The gut microbes are able to synthesize essential amino acids and vitamins, boosting immune function and decreasing disease permeability. However, with the decrease seen in gut microbes in the winter season, one can conclude that the polar bears may be more susceptible to diseases due to the decline seen in gut health and digestion. Low bacterial richness, as mentioned earlier, further links adiposity due to indigestion occurring, leading to slower metabolism, and a higher inflammatory status. Gut microbes can be further linked to providing essential sulfur amino acid, which increases the synthesis of glutathiones by acting as a precursor. The amino acids further assist with functions of detoxification and mucosal integrity maintenance, all of which assist immune function. The transit of food through the stomach, small intestine, and colon is crucial for digestion and absorption of nutrients. Digestibility of food can be affected by food composition and passage rate, where a faster rate reduces digestion. Polar bears rely on their carnivorous diet because their body demands a high caloric intake, and they digest fat 97% more efficiently than proteins, which is 84%. Which is why you can see in the table, the gastrointestinal tract time, which is GTT, is faster when the lipid extract percentage is more compared to the protein extract percentage, as protein is more time consuming to digest than lipid or fat content present in the blubber. This is why polar bears prefer a lipid-rich diet, as they have preferred affinity for fat content over protein.
Now, with this experiment in mind, we can acknowledge the fact that climate change and its warming effects may disrupt polar bear digestive systems. Homeostasis involves maintaining steady internal, physical, and chemical conditions within the body, and this experiment highlights two of its main contributing factors: body temperature and digestion. Exploring the relationship between the two is important for many reasons. Firstly, increasing rates of climate change have proven to be detrimental, and polar bears are one of the many populations that continue to be negatively affected by it. The severity of these conditions must be addressed through temperature-dependent experiments such as this one in order to proactively work against the ramifications that are capable of getting worse. Many authors have acknowledged the outcome of changing temperature on a diversity level. However, a large void exists in regards to the physiological and behavioral detriments existing concurrently. Approaching the issue of climate change on the level of polar bear tissue and organs makes room for interpretation of these results on a population level within the same species. We can use the results to predict future behavior of polar bears in both worst and best case scenarios, and even possibly save these vulnerable species from extinction. Among other things, warming effects on digestion should be observed more closely because the topic is extremely rare, hardly existing among scientific literature. Understanding endangered marine mammals on an integrative level will produce results that are fundamental for future studies to come. Therefore, its importance is one that should be emphasized.
As with any scientific study, this study contained its fair share of limitations and possible sources of error. One major limitation of the study was the captivity of the polar bears. These polar bears were held in a lab without any of the stressors they would otherwise face in the wild. The captivity of the bears allowed them to get as much food as they want without the struggle of finding it in the first place. In the wild, a polar bear would get lucky to secure kills once every 5 days, so hunger is a major stressor. Without the added physical activity needed in the wild, it is highly possible that the fairly inactive lifestyle of the captive bears may influence the hunger of these bears and thus the quality of food they eat, as well as other factors such as obesity, which was discussed earlier. Another major limitation of the study is the lack of account of natural cycles. Though these bears are subjected to normal daylight rhythm, other natural cycles are omitted from this study, which may influence final results. For example, polar bears tend to remain largely inactive during the summer months, as discussed previously, and this is because of the evolutionary selective pressure that food is less available in the summer months, so the bears prefer to exert as little energy as possible. This behavioral trait of polar bears can also influence the physiological response of the bears during the course of the study, where their internal circadian clocks and other natural cycles are still synchronized to the normal external environment that they're used to. These factors have the possibility to largely influence the final result of the two experiments that were conducted. All in all, these two studies aim to bring to light on how changes in temperature can lead to physiological changes in polar bears, not just on the digestive system, but on an organismal level as a whole. Furthermore, it is encouraged that the techniques learned from this study be applied to other organisms and on other systems. There are several areas of research that can be delved into from this study as a stepping stone. Mentioned previously were studies looking into the indirect influences of external temperatures on digestibility through enzymatic function, as well as changes to lipid composition based on dietary content and how these can influence thermoregulation. The effects of external temperatures on polar bears can also be extrapolated to a populational level, as mentioned before, not just a cellular or a tissue level. Polar bears face a grave future as their home is being melted away, leaving many bears starving and adrift in the Arctic Ocean. In a world where climate change is a global issue and potential threat for hundreds of thousands, if not millions of species, it is imperative that further research should be conducted regarding this field. And this concludes our presentation. Thank you so much for watching.